Today, my poster topic is can the COVID RNA vaccine trigger the autoimmune responses? So, for that, I want to start the introduction part. The computational proteomics uses algorithms and models to investigate the active predictable proteins. The main research in this area is often devoted to the development of vaccines and the development of computer-assisted drugs. Strong targets in this method of autoimmune body count calculations, which are the result of a broken immune system where they are unable to distinguish between self and non-self, leading to attacks on their structure. Here, the proteins and DNA. Although we do not aware of the particular etiology or of autoimmune diseases, there are a variety of aspects that are believed to be the cause of autoimmune diseases, including genetic predisposition, natural causes like viral infections, including gut microbiota, functional viruses, and gluconomous infections, along with body and environmental agents, hormonal features, etc. Coronavirus correspond to a large group of viruses that mainly affect people through the zoonotic transmission. Some patients experience severe phenomena with sepsis leading to respiratory syndrome with the respiratory failures and sometimes accompanied by hyperferritinemia and multiple organ failure, nerve failure, or any kind of heart problem, etc. Some researchers are saying that it is leading to the death also. It has been suggested that the shared pathogenic mechanisms and clinical features and radiological radiation between inflammatory diseases and COVID-19 may suggest the SARS-CoV-2 might be the factor of development of acute autoimmune disease. So this is the reason we are doing the research here to identify whether the COVID-19 virus trigger autoimmune disease or not. The anti-DNA antibody is found in many patients associated with autoimmune disease. However, the chemical composition and processes under autoantibodies remain poorly understood. Some researchers were able to differentiate between anti-DNA and only a small amount of binding sites of anti-DNA antibodies. Sequencing of weight makers and X-ray crystallography are expensive and time consuming also, and it requires expertise for each cell. Computer methods, however, which are a model of existing data for making novel predictions, can work to reduce the field of nominees who may be tested in the lab. Classical machine learning approaches often face difficulties in processing raw data due to the need for handcrafted features. Reversely, deep learning methods can derive practical features directly from the data. However, for more than 50 years, neural networks were not an option for machine learning, and the advancement of the powerful computer hardwares and many fast processors like GPU, the combined with a lot of training data, uh, recently made the deep neural network or DNN perform better in many machine learning algorithms. So we have some recent machine learning algorithms worked on this research. Clear et al. Have used the short-term memory model or LSTM model directly trained on FASTA primary sequence to predict the protein binding in DNA binding hydrolytic antibodies. They were also used the CNN model applied to the same dataset, although the CNN model outperformed uh, on the LSTM model. So they researched mainly on the protein binding or RNA binding, how we can calculate the RNA binding, but they did not distinguish between the human RNA binding and viral RNA binding, and they did not try to um, calculate the antigenicity of both on based on the bindings. So our approach is based on the machine learning algorithm, preferably the deep neural network. We are trying to create a software platform through which we can uh, identify and distinguish the RNA bindings for both virus and human, and after that, based on that distinguishing, we can calculate the antigenicity for both to summarize or conclude that whether the RNA vaccines trigger the autoimmune response. So here are some of my references listed, and that's all. Thank you.